Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing CSIR Net JRF Life Science question paper part C questions. This is of shift 1. It was conducted in July 2024. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers being discussed of CSIR Net JRF Life Science, you can go to my channel Omni Bioentrance. In the channel playlist C for CSIR Net JRF, in that folder you will find all the question papers. Now coming to the first question. Given below are four topology diagrams corresponding to different proteins. N and C denote the N-C terminal ends of the protein chain. Which of the following statement is correct? Now if you see A from N, from C terminal to N terminal. Here also in C, from C terminal to N terminal. Between these two central ones, from N terminal to C terminal. Here also from N terminal to C terminal. From C terminal to N terminal. From C terminal to N terminal. And the orientation is also same. One is like this. Here also the arrow is pointing that direction. This also same way. This also same way. This also same way. But in D, if you see, their orientation is not the same. Okay. But B, it is entirely different turns. So the answer here will be A and C are of the same fold. 4 is the answer. Then next one. The plaque morphology of the wild type and R2 mutants of T4 bacteriophage following infection of different E. coli strains is summarized below. So this is the T4 phage strain, wild type and R2 mutant. This is E. coli strain B and K. Now wild type mutant, it forms in B strain of E. coli small and ragged plaques. In K strain again small and ragged plaques are formed. But R2 mutants in B, it forms large and round plaques, whereas no plaques will be obtained on R2 by the in K strain. Now following experiments were carried out. Co-infection of the two independent R2 mutants on E. coli K strain resulted in several plaques, all being small and ragged. That means they got the R2 mutant upon infection on E. coli K strain gave rise to the wild type plaques, that is small and ragged. That means the R2 mutants complemented each other. Okay, they complemented each other, suggesting that is their mutations in the R2 mutant got complemented with each other, giving rise to wild type plaques. Now experiment 2. E. coli B strain was co-infected with the above R2 mutant. So this is the B strain. It was co-infected with the above R2 mutants. These R2 mutants were infected on E. coli B strain. T4 phages from the resulting plaques were used to infect E. coli strain K. Now what they did, the T4 plaques, whatever they were obtained from E. coli B strain, was used to infect E. coli K strain. What did they obtain? Few plaques were obtained which are small and ragged. So only very few wild type plaques were obtained, showing recombination. So recombination has taken place in E. coli B strain, and upon infection of E. coli K strain with these recombined R2 mutants, you got only few plaques which were small and ragged, showing recombination. So based on the observation, the following statements are made. Experiment 1 indicates that two mutants are allelic. That is incorrect. They should be non-allelic to complement each other. If they were allelic, that is they had mutation in the same gene. Suppose there are two genes A and B. R2 mutant is having mutation in A gene. The other R2 mutant is having mutation in A gene. Then will they complement each other? No, they are allelic. So they will not complement each other. Now suppose R2 mutant, first one is having mutation in A gene and the R2 mutant, second one is having mutation in B gene, then A and B can complement each other and result in wild type phenotype of the plaques. So they are non-allelic. They should be non-allelic to complement each other. So A is wrong. So wherever A comes, it is wrong. So 1 is wrong, 3 is also wrong. Experiment 2 indicates that the wild type T4 fudge that infected E. coli K strain resulted from recombination event. I already explained that. Then in experiment 2, if the T4 fudge is isolated from E. coli strain B was used to infect E. coli strain B, all plaques would be large and round. No, only R2 mutants will give large and round plaques. Wild type will give small and ragged plaques. So C is also incorrect. So the answer here is option B only. Now, given below are figures representing 4 different situations. Examples of genetic and environmental effect on the plant height in 2 varieties of plant species, that is G1 and G2. In figures A, C, D, solid and dashed line represent the mean value of plant height for the 2 varieties G1 and G2. In figure B, the solid and dashed lines overlaps. Okay. Now, given below are 4 statements explaining the 4 figures. Plant height is influenced primarily by the genotype of the 2 varieties. Now, what you have to do, you have to match. Okay, match the following. First one. Here what is happening? Temperature doesn't have effect on the plant height. Only based on the genotype there is variation. So genotype is influencing, whereas temperature is not influencing. Genotype is influencing, that is why you get different heights for G1 and G2. So plant height is influenced primarily by the genotype of the 2 varieties is by A, so A1. So 3 and 4 not correct. Then this graph, here G1 and G2 overlap, but as the temperature increases, plant height also increases, but it is not dependent on the genotype. So variation in the plant height is influenced only by the temperature and genotype has no effect, that is B. So B2, that gives your answer as option 1. Then third graph, temperature is influencing. As temperature increases, height also increases. There is variation in the G1 and G2. So temperature and genotype collectively have an additive effect on plant height. So C is 3. Now in fourth one, if you see, in case of G2, temperature increases, plant height decreases. In case of G1, temperature increases, plant height increases. So both genotype have an effect on and environment have an effect on the plant height, yes, with the 2 varieties responding differently to the environment. 4. D4. So that is correct. So A1, B2, C3, D4. Then next one. RPMI and DMEM supplemented with serum, antibiotics, glutamine and phenol red are routinely used for tissue culture of human cells in carbon dioxide incubators. In addition, sodium bicarbonate and HEPs are used as buffering agent. The following statements are made about the media. Now, why do you add phenol red? Phenol red is an indicator. Okay. Now, in the neutral condition, it is orange red in color, okay, and it becomes yellow when it is acidic, and it becomes red or pink when it is basic. Now, how does this culture medium convert to acidic, convert to basic condition? Now, in a rapidly dividing cell, the culture metabolizes the nutrients and produce lactic acid. So, the medium become acidic. So, the phenol red changes from orange red to yellow, indicating drop in pH. Now, when it turns basic, because when there is when cells are not metabolizing properly, then the medium becomes alkaline and it turns pink to red, showing that there is increase in the pH. That is what phenol red is doing. Now read the statements. While 5% carbon dioxide is optimal for cell cultured in RPMI and the optimal carbon dioxide concentration for DMEM is 7.5 to 10%. That is correct. So A is correct. So wherever A comes, it is correct. So B is not correct. 4. Okay. HEPS is necessary if cells are to be kept outside the incubator in room air for long period. Yes, it is a Zwitter ionic buffer and it maintains pH at room air conditions. Because carbon dioxide is lower than that in the carbon dioxide incubator. So B is also correct. So A and B are correct. That means 1 is your answer. Okay, we will see C and D also. What is the mistake there? Let me see whether A and B is the right answer. Yes, A and B is the right answer. Okay, now NaHCO3 is necessary if cells are to be kept outside the incubator. Kept outside the incubator is for HEPS, not for NaHCO3. So C is wrong. When cells grow rapidly in the culture medium, phenol red will turn medium pink to red. No, it becomes acidic because lactic acid is accumulating there and it will turn phenol red to yellow, indicating there is a drop in the pH. So D is also incorrect. So A and B are the right answers, whereas others are not correct. Then given below are statements on transgenic plants. Transgenic plants generated using a transformation vector with the CMB35 as promoter, GUS35 as PA cassette can show variation in the expression level of GUS independent of transgenic events due to difference in strength of the promoter. No. It is not due to difference in the strength of the promoter used to express the GUS gene. It is because of the positional effect of transgene insertion. Sorry. Now what is this positional effect of transgene insertion? It is that the transgene can get inserted into the genome randomly. And because of this, the gene can be expressed at different levels. Okay, so A is not correct. So 1 is not correct, 4 is also not correct. A transgenic plant containing 2 insertions of the transgene cassette as inverted repeats in tandem. Remember this, inverted repeats in tandem. That is, one will be in this direction. Just like your palindromes, this will be in this direction. And in tandem means they are arranged one after the other in this way. In this way they are arranged one after the other, would segregate in a 3:1 ratio for the transgenic phenotype on back crossing the transgenic plant with the untransformed parent. Now when it is inverted repeats in tandem, it means that the gene is going to be silenced. So how will you get all these ratios? You are not going to get there because silencing doesn't show Mendelian inheritance. So B is not correct. Anyway, from option 1 itself, you know B is not correct. Okay, C, D, we will see. A transgene containing a potential polyadenylation signal in its coding sequence. Okay, fine. would generate full-length mRNA. No, in the coding sequence means in your exon. Now, if it is having a polyadenylation signal in the coding sequence, it will not generate a full-length mRNA transgene. It will be a truncated mRNA transgene. And it might result in premature termination of transcription. Okay. So C is not correct. A gene pyramiding experiment to bring together two transgenic traits by crossing independent homozygous single copy transgenic lines for each trait would produce plant homozygous for both the transgene in the F2 generation. Now, in gene pyramiding, if two independent homozygous to bring together two transgenic traits, okay, two independent homozygous single copy transgenic lines are crossed, the F1 generation will be heterozygous for both the traits. When the F1 generation is selfed, you get the F2 generation, which will segregate in Mendelian fashion, where some plants will be homozygous for both the transgene. So D is correct. So the answer here is option 3, that is D only. Then 4 group of students A to D were asked to determine whether memory cells generated in mice immunized with ovalbumin in complete Frounce adjuvant could mount a secondary antibody response, recall response to ovalbumin in vitro. The group did the following experiment. Now, what is this secondary antibody response, primary antibody response, etc.? Now, primary immune response is when an animal is exposed to an antigen for the first time. Here it is ovalbumin. It will produce antibodies to neutralize this ovalbumin because it is an antigen. Okay. Now, these antibodies, that is B cells, they will differentiate into plasma cell secreting antibodies and memory B cells. The memory B cells will go and store themselves in the spleen cells and they will remember this antigen, okay, for future encounters. The plasma cell will, which is secreting the B cell antibody, will do their job at that time, that is primary infection, of throwing away the antigen from the body. Now, what is secondary immune response? Now, again, if you inject the rabbit with the same ovalbumin, what will happen is the memory B cells will get activated. Yes, I know this antigen. And they will start to secrete plasma cells and memory B cells again. And these plasma cells will go and neutralize the ovalbumin. And it is more faster than primary response. Now, what has group A done? Group A has harvested the serum from the mice, loaded it on ova-coated ELISA plates and showed that IgG, IgA, anti-ova antibodies were present. Now, they have shown the presence of antibody in the serum and this doesn't tell anything regarding the secondary antibody response or the recall response. That is, memory cell response will not be shown here. So, group A is not correct. Group B students harvested long-lived plasma cells. Long-lived plasma cells are not your memory B cells. They continuously produce and secrete antibodies that neutralize the antigen upon reinfection. They are not your memory B cells. And when these long-lived plasma cells, they cannot defend the pathogen, then your memory B cells will be stimulated to mediate the defense. So B is also not correct. Then group C students infected an epithelial cell line with the virus. Virus is not going to show you antibodies. It is going to show you cytotoxic T cell activity. So C is also not correct. What is remaining? Group D. Stimulated spleen cells. Yes, spleen cells I told you will have memory B cells from the mouse with the ovalbumin for 5 days. So they stimulated the memory B cells for 5 days with the ovalbumin. So memory B cells, that is your secondary antibody response or the recall response got activated, and they showed anti-OVA antibodies in supernatant by ELISA method. So they will show secondary antibody response. So 1, 2, 3 are not correct, 4 is correct. Then next one. A researcher used CRISPR-Cas9 system and observed a different type of mutation in the 2 alleles of a target gene in a T0 transgenic plant. These mutations are designated as follows. I told you this is allele gene variant of the same gene. Are called allele. It can be A, capital A and small a. They are alleles. These mutations are designated as follows. The observed mutations can be classified as. Now what is monoallelic mutation? Occurring in one of the 2 alleles of the gene. Mutation occurring in one of the 2 alleles of a gene is called as monoallelic mutation. What is biallelic mutation? Mutation occurring in both the alleles of the gene, and it can be classified into biallelic heterozygous mutation, biallelic homozygous mutation. Now both the alleles have the same type of mutation, that is they have the same addition, same deletion, then it is biallelic homozygous mutation. Now if they have different mutation, that is one allele is having addition, another allele is having deletion, then they are heterozygous mutation. Now allele 1 is having addition of a nucleotide, allele 2 is having deletion of a nucleotide. So the observed mutation is classified as biallelic heterozygous mutation. Answer is option 2. Then next one. The theoretical resolution limits of a fluorescent microscope in a 200 nanometer super resolution microscopy has been developed to address this limitation. Given below are super resolution microscopy methods in column X and they are principal in column Y. This you have to learn from your theory, but I will give you the answer here. Structured illumination microscopy. That is, the specimen is illuminated with a pattern of light and dark strips to generate more fringes. A2. 2 is there, 4 is there. So 1 is not correct. 3 is also not correct. Stimulated emission depletion microscopy. That is, focused excitation laser point is surrounded by a donut shaped depletion beam. Photoactivated localization microscopy utilizes variant of GFP that is activated by a wavelength different from its excitation wavelength. So answer here is option 2, that is A2, B1, C3.